Hey, I'd like to welcome you all to the IRES Symposium. We've had a break. Last year, we could not do the symposium, much to our chagrin. We were in the middle of the first wave, and now we are smack in the middle of the third, but we know how to do things better. So we have a symposium this year. But before we get into the speakers and their supervisors, I'd like to do the land acknowledgement. I'd like to acknowledge that UBC's Vancouver Point Grey campus is situated on the traditional, ancestral, and unceded territories of the Musqueam people. I would also like to acknowledge on your behalf that you're joining us today from many places, near and far, and acknowledge the traditional owners and caretakers of those lands. With that, I'm going to hand over the proceedings to Professor Hadi Dalatabadi.
Thanks, Milind. So not only were we not able to hold this last year, but the year before, Terry wasn't there. And for many years before that, we collectively, faculty and students, kept nominating Terry for Killam Exceptional Advisor recognition. And the university, being the wise institution that it is, kept picking somebody from nursing with 1,500 students just about to retire. And so we were thwarted 4 years in a row. And so while it's taken a long time, Terry, for us to express just how much we appreciate you and how much we wish this was done in front of all of the university community, we've just decided that no further delay is possible. God knows if I'll be here next year to do this for you, but here is a little envelope that we have put our collective thanks in, and I'm handing it over to you. Would you like to pick it up? At your end, this is the latest service that Zoom provides, Terry. Terry?
I think she's frozen.
Where are you?
Thwarted yet again.
Man.
Oh, God, she's gonna come back. She's gonna come back.
Yeah. I think she decided to leave us. The
No,
whole
she'll
idea
come back.
was that
She'll
I would
come back.
pass the envelope to Terry and she would pick it up and open it up and see what it is.
Maybe she's on the way to your house, Hadi.
No, we had so much trust in Canada Post that Jillian delivered it to both of us. Otherwise, it could have been another year before we got it. So, hey, look, let me just, you know, this is recorded, so she'll be able to get it anyway, right? T. Lim and so many other of Terry's students just couldn't be.
We just lost connection. Okay, get back on.
Yeah.
Okay.
More supportive of her and our attempts at nominating her for wide recognition. And it's just been a delight because if you read the letters, they include faculty who consider Terry to have been an incredible mentor to them, myself included. Milind stuck his hand up too. So yeah, it's a book that contains letters of appreciation from a subset of those who Terry has mentored. And I'm sure that if we'd kind of made it even more public and even more recent, there would be even more people who would be delighted to have their letters included in the package that is in Terry's hands. Could you please open up your package now?
Okay, I actually seriously lost connection. Can you believe it? Thank you, Hadi. I know exactly what you're talking about now. Appreciation from a subsample of beneficiaries. Can you see that? Hadi, can you explain it? I've missed most of what you said, but I get the gist for sure. And thank you.
It's simply a case of all of us wanting to thank you in as concrete a way as we could, in a time in which we can't be together, but can't be delayed any further. Anyway, Terry, thank you very, very much on behalf of the broader community and many of your students and faculty colleagues who consider you to be a truly invaluable mentor to all of us.
Thank you. Thank you very much. I feel kind of silly. That seems a long time ago now, but thank you, everyone, seriously. Much, much appreciated. And there are a few things that I love more than that opportunity that is IRES, and to work with all of you and so many of you. So, and if I say anything else, I'm just going to completely embarrass myself. So I'm going to turn the microphone off.
Good. Okay, so let's start the talks and proceedings. I'm going to hand over the mic to Rashid to introduce Annie Mejaes.
Yeah, thank you very much, Milan and Terry. From the lower floors, we send our congratulations to you. You've been a wonderful colleague in so many ways, so that's great. Okay, I have the pleasure of introducing to you Annie Mejaes. And Annie, as many of you will already know, is pursuing her Master's of Science degree in Resources, Environment and Sustainability, of course, in our department here at UBC. She's a member of our Fisheries Economic Research Unit under my supervision. And today she's going to share to you actually a thesis. I really think she's the first one to seriously look into the issue of subsidies to the aquaculture sector. People have done little pieces here and there, and we've spent a lot of time looking at subsidies to the wild fisheries, and this is the first one. So I'm really delighted to introduce you, Annie. And before you came to us, you had a lot of experience in conservation, environment. You know stuff in geospatial work and analysis. You work for government agents, so you were quite well experienced before you came. You've worked in Hong Kong, in Chile, in the USA, so you are well versed, and I'm so happy to have you as a student. And now on Monday she's defending. So if you want more stuff, just, oh my God, it's that thing again, COVID. You cannot even join that defense. So anyway, over to you, Annie, and good luck with your presentation.
Thank you very much, Rashid. And I will share my screen. Can everyone see my screen?
Yeah.
Great. So good afternoon, everyone. Thank you all for being here. Today, I will be talking about my master's research on aquaculture subsidies. As a quick background on why aquaculture matters, human consumption of seafood is increasing. Not only is this a result of an increasing population, but since 1960, our consumption of fish per person has doubled. Yet, our fisheries are stagnating as they are reaching maximum capacity. So aquaculture is dramatically increasing to supplement the demand of our seafood. So what are subsidies? Subsidies can be defined as an intervention that includes an economic value, which is often provided by a government. Subsidies are present across food production sectors. In agriculture, an estimated $700 billion of subsidies are provided yearly. As you can see, fishery subsidies are equal to the value of 1 in 3 fish. So what about in aquaculture? Governments are also providing subsidies to the aquaculture sector because aquaculture can potentially contribute to food security and economic development. While regional and country-specific studies do exist, no one has specifically measured or classified aquaculture subsidies on a global scale. The goal of this study is to begin to fill this gap. After all, for aquaculture to provide a stable and continuous contribution to our food system, not only is the sustainability of the sector crucial, but also how our government subsidize the sector should be carefully considered. To address this gap in knowledge, my research answered 3 main questions. What aquaculture practices and subsidy types are beneficial, ambiguous, or harmful to marine ecosystems? What are the aquaculture practices and amount of subsidies in the top producing countries? And finally, do any patterns emerge? What are the policy implications? Just as a side note, my work focuses primarily on mariculture, which is aquaculture or brackish water or marine, which is aquaculture for brackish water or marine species. So to answer each of my questions, I designed a classification system, created a database, and analyzed the data respectively. Next, I will dig deeper into the design of the classification system. So where do I start? As there is no classification dedicated to aquaculture subsidies, I look to agriculture and fisheries, which both have known subsidies and well-defined classification systems, and also share similarities with aquaculture. I examined both sectors to understand their common traits and the basis for their classifications, which helped to shape how I classify aquaculture systems and aquaculture subsidies. What I realized is that aquaculture very much sits on a spectrum between both fisheries and agriculture. Existing on this spectrum, it was evident that there are a wide variety of aquaculture systems and that I would need to account for this diversity in my classification system. Aquaculture systems may be on land, in the water, may or may not require fish feed, may be native or exotic species, the list goes on and on. To further dig into aquaculture and its risks, here is a basic conceptual diagram I drew. The first component distinguishes between the two main sources of farmed fish, which is either directly from wild fisheries, capture-based, or hatcheries. The second component provides examples of the main aquaculture systems that are classified by their locations and stocking density, among other variables. The third component represents the post-harvest value chain. Now, subsidies can be linked to every single part of this conceptual model, even the inputs and outputs. Given the diversity of aquaculture systems highlighted in components 1 and 2, the environmental risks posed by aquaculture systems differ in type and magnitude and may include effluent discharge, fish feed, habitat deterioration, and escapees. Subsidies can also impact an aquaculture system positively or negatively with respect to the environment. Wherever possible, we want to limit the environmental risk and promote the sustainability of aquaculture if we are going to be providing subsidies. So for my classification system, I incorporate both the aquaculture production systems and their environmental risks, in addition to the subsidy categories, before bringing both segments together. The comparison of fisheries and agriculture informed how I categorized aquaculture production systems. I identified measures that nested aquaculture types. So any given aquaculture system could be characterized as an aquaculture type for every measure. For example, one measure is if the species is exotic or native to the country. To determine the environmental risks of each aquaculture production system, we used risk factors on a scale of 0 to 1. Relying on literature review and existing life cycle assessments, the scoring was based on the relative impact of the aquaculture system on the marine environment. Existing fisheries and agriculture classification systems provided the context to propose the subsidy categories, which were iterated upon when I started to collect data on subsidies. Finally, I brought the two components of the classification system together to understand how subsidies are impacting aquaculture systems and their risks. As there isn't significant literature that exists on it, colleagues also provided input to support this final section. Now, this is a table that I use to classify aquaculture systems by incorporating the measures across systems. In this table, you are seeing just 2 of my 6 measures. The risk factors highlight the relative risk associated with any given aquaculture type, where one poses the greatest risk. So for instance, extensive aquaculture requires no fish feed and so poses less risk to the marine environment than intensive aquaculture. As another example, while hatchery-based aquaculture uses broodstock to cultivate juveniles, capture-based aquaculture relies on natural fish populations for seed, eggs, or juveniles. So for this particular measure, capture-based aquaculture has a greater impact on fish stocks. We are careful to ensure that this table encompasses universal qualities of aquaculture systems that are agnostic of technological development to account for future innovation in the sector so that it does not become out of date. Next, the subsidies themselves. Similar to classification systems across agriculture and fisheries, I propose a typology parsing the objective of the subsidies. So we classified subsidies based on 10 overarching categories. By classifying subsidies initially according to the subsidies objective, it makes it easier to analyze the impact of each subsidy type on the marine ecosystem. Finally, we bring both components together. For each of the 10 subsidy categories, I explored how subsidies may impact, for better or worse, the risks posed to the marine environment. In addition, a subsidy can encourage aquaculture practices that may not currently be present or widespread in a country, such as the introduction of exotic species. Here, I provide an example of 3 subsidy categories and how each of them may exacerbate or lessen an aquaculture system's impact on the marine environment. Aquaculture assistance may promote overcrowded systems or may restrict assistance and capacity building initiatives to sustainable aquaculture practices. While infrastructure funding may subsidize infrastructure in sensitive ecosystems, it may also promote closed aquaculture systems that would minimize pollution. Research and development funding, on the other hand, may promote exotic species or could reduce the demand for wild fish stocks as an input by encouraging the development of alternatives to fish feed. So to wrap up this section, government expenditure, including subsidies, is generated primarily from the tax revenue of private citizens. So it is important to conduct due diligence. To do so, we have attempted to understand the role that subsidies play on the potential risks of aquaculture production systems on the marine environment. Also, as a rapidly evolving industry, continued improvements in aquaculture are expected and can lead to an increase in the technology's resource efficiency. Thus, while aquaculture types may hold true, the environmental risks associated with each aquaculture type may shift over time. For example, intensive aquaculture may no longer require substantial quantities of wild fish in the future. So putting the classification system into practice, I will move on to the 4 case studies. I selected these 4 countries because they represent the largest mariculture producers globally. Together, these 4 countries represent over 70% of mariculture production, each with distinct socioeconomic, geographic, and government contexts. China by far is the world's largest mariculture producer. China also has the highest diversity of species, followed by Indonesia. Norway and Chile, which were comparatively latecomers to aquaculture, primarily produce Atlantic salmon for export purposes. For each case study, I determined the predominant species and aquaculture production systems, as well as classified and estimated the subsidies. To categorize aquaculture systems, I identified specific characteristics necessary for the cultivation of each species, using peer-reviewed literature and permit information. I also determined the potential risk that each country's aquaculture poses to the marine environment using the proportion of aquaculture types by weight and the corresponding risk factor. To categorize and estimate aquaculture subsidies, I gathered information by subsidy category from publicly accessible resources. I also recorded subsidies that could not be quantified, but that did exist. Finally, I analyzed how subsidies are employed within a country's aquaculture sector and what types of risks are being exacerbated or lessened. The following equation was used to calculate the percentage of relevant subsidies that is likely to impact a given risk. So China's mariculture sector has rapidly grown and is predominantly comprised of bivalves. These 6 species represent over 75% of total mariculture production in China and are mostly grown on long lines or in mudflats. Bivalves are mostly cultivated in hatcheries, but certain species, like the mussel and clams, are also collected from the natural environment. The one crustacean, the white-legged shrimp, is an exotic species and is the only semi-intensive species on this list. As you can see, an estimated $3.9 billion are provided in subsidies. The vast majority are for construction and renovation, as China has invested significantly in marine ranching zones and deep water cages. In addition, there are subsidies that could not be quantified and are not included in our estimates. China has piloted subsidized mutual insurance schemes, and to promote the growth of the sector, only 50% of an aquaculture company's profit is taxable. Moving on to Indonesia. Indonesia has been focused on expanding its aquaculture sector for livelihoods, local food security, and export purposes. In Indonesia, the farm species are predominantly cultivated in semi-intensive brackish water ponds, relying on fish feed as well as the natural environment. All of these species also have a closed life cycle. The only exotic species is the white-leg shrimp. Approximately $987 million is provided in subsidies to the sector yearly, with over 90% provided for fish farmer assistance through a business development program. Approximately $45 million are provided specifically to shrimp and milkfish production to support exports and local seafood consumption. Indonesia has also provided a significant amount this year for COVID-19 support, for the COVID-19 pandemic to fishers, farmers, and processors. There are also unquantified subsidies, including tax credits for local and foreign investment and an insurance scheme. Moving on to Norway, in 2018, Norway produced 52% of the world's farmed Atlantic salmon. Both species, Atlantic salmon and rainbow trout, represent over 99% of Norway's aquaculture production. Both species are also cultivated in the same manner. As smolts or juveniles, they are raised in freshwater environments, most often in flow-through systems, before being transferred to open net cages. Both species are also carnivorous species and rely on fish feed as an input. An estimated $282 million of subsidies is provided to the aquaculture sector annually in Norway. Within research and development, with 60 to 70% of the budget is allocated to salmonid research. Norway has also started to subsidize development licenses for offshore aquaculture. In addition, unquantified subsidies include subsidized licenses for teaching purposes and land-based facilities. Finally, Chile's aquaculture sector is also almost exclusively mariculture and is focused on high-value exports. Atlantic salmon, coho salmon, and rainbow trout are farmed in a similar manner to those in Norway. The Chilean mussel, on the other hand, is a local species that represents 25% of the country's mariculture production and is grown extensively in the marine environment, although the species relies on seeds collected from the wild for farming. There are an estimated $115 million in subsidies provided to the mariculture industry in Chile annually, with nearly half provided to management. Over 10% is also provided to the rural development of aquaculture, as Chile is making an effort to improve the productive capacity of small-scale aquaculture nationally. The unquantified subsidies include a tax concession if a company invests in research and development, and also a tax exemption for aquaculture companies located in a specific super free trade zone in the south of Chile.
Annie, you have 5 minutes remaining.
Okay. Combined, the four countries contribute approximately $5.24 billion in subsidies to the mariculture sector in 2020. Similar to recent estimates on marine fishery subsidies, the Asian countries dominate in terms of total amounts provided. In addition, Indonesia, followed by China, have the largest subsidy intensities. However, the subsidy intensities are quantitatively significantly lower than those of agriculture and fisheries. But the estimated subsidies and subsidy intensities for the case studies are underestimates, as evidenced by the unquantified subsidies outlined for each country. This table here shows you 3 out of the 6 measures. The risk index is calculated based on the proportion of the subsidy and a country's risk factor scoring that relates to a specific measure. A risk index of 100 signifies that the aquaculture measures possesses the highest relative risk possible, and that all of the subsidies provided to a country's aquaculture sector impact those risks. The arrows indicate whether the subsidies primarily lessen if it points downward, or worsen if it points upward the measure. If the arrow is flat, then there's a combination of subsidies at play. Norway and Chile both have high risk index scores for the first 2 measures, as both countries primarily cultivate salmonids in intensive open systems. In both instances, however, there's a combination of subsidies that are promoting as well as subsidies that may lessen the risks associated with this farming. Looking at the life cycle of production, all of the countries apart from Norway rely on capture-based aquaculture for a proportion of their aquaculture development. However, only China has a significant risk index score for the measure because Chile and Indonesia dedicate minimal subsidies that impact capture-based aquaculture. However, capture-based aquaculture needs to be managed and monitored effectively to limit its negative impact on wild populations. With strict regulations, subsidies can provide sustainable aquaculture that supports livelihoods, the economy, and food security. To do so, each country can redirect certain subsidies that exacerbate impacts on the marine environment. I will provide you with a couple examples from each country. China is the only country with fuel subsidies dedicated to aquaculture. These subsidies should be eliminated. In addition, as China's 13th five-year plan is promoting the sustainability of aquaculture, it would be appropriate to include environmentally focused requirements to the corporate tax subsidies, such as effluent discharge limits or land use requirements. Indonesia has implemented insurance in case of disease outbreaks. In addition to relying on insurance, the country has set density limits and limits insurance to farmers who abide by the policy. Indonesia's new subsidies to encourage shrimp production could also be restricted to native shrimp species rather than also subsidizing an exotic species. Norway provided a tax credit to companies that engage in research and development, but there's no requirements for the type of research, so it could very easily be self-serving. The research should be monitored or specified by the government. In addition, many producers are taking advantage of free specialty licenses for teaching or demonstration purposes to increase production. This is effectively a loophole that can be closed. And finally, Chile has free trade zones where the majority of the country's salmon farming and intensive species is located. These zones do not have any taxes and actually provide money to the companies. This is not benefiting small-scale farmers, but multinational businesses. Also, as the government is promoting small-scale farming and Chilean mussels, subsidies can be provided to close the life cycle, establish hatcheries, and provide seeds to limit the collection of seeds from the wild. So my final thoughts. Apart from Indonesia, the other three countries have begun to prioritize the sustainability of the sector and their natural resources, with less emphasis on production volume. Yet only Norway and China have specifically dedicated subsidies to environmental concerns. Moving forward, countries need to ensure that their policies align with their subsidies. It is also important to analyze the subsidies in the context of the aquaculture systems because Norway and Chile are more export-oriented, producing higher value species, whereas China and Indonesia have a larger percentage of small-scale aquaculture and are more concerned with supplying the local population. This impacts what type of subsidies each country provides and the aquaculture practices that are promoted. Finally, this work highlights that subsidies need to be carefully considered within the aquaculture sector. In particular, it is important to understand that environmental risks associated with different production systems to avoid exacerbating these risks via subsidization. So we need to continue building upon this work to measure and classify subsidies globally. And as a super far-reaching, shoot for the stars type of goal, maybe one day someone would be able to build upon this research to be able to influence policy, to direct subsidies towards aquaculture practices that pose less risk to the marine environment. Thank you very much. Thank you, guys.
Thanks, Annie. Is there questions? How are we?
Questions? Am I supposed to run the questions or Annie will do it?
Supervisor runs the questions.
All right, let's go. I see that Sara has a question. Your hand is up here on my screen. Go ahead, please.
Yes, hi everyone, and hi Annie. Thank you so much for a really interesting presentation. I was just wondering if you included any anything about offshore aquaculture in your analysis in federal waters? And if so, if you distinguish between offshore and nearshore aquaculture when looking at the environmental impacts, just because I know that offshore aquaculture, at least people tend to assume that that has a lower risk for the environment due to the distance from the sensitive coastal ecosystems. Although I know that depends on many things, including the species that are farmed. And then just building off of that, I was also curious if you know if there are any subsidies specifically for offshore aquaculture, like if this is something that governments are supporting. And I asked just for some context, I know that in the US, it's a really controversial issue right now. So I was just curious if that was the case elsewhere.
Yeah, so to answer your second question first, in China and Norway, they're both providing quite substantial subsidies for the development of offshore aquaculture. In Norway, it's offshore aquaculture that is also semi-closed, so it will have so it won't be directly open and interfacing with like the marine environment. Instead, there may be some type of like plastic sheet or tube or whatnot. And then in China, they're also building like, they call it like deep water cages. And the idea is that I think what they say is like dilution is the solution. And so because it's offshore, then technically there's like less marine life per area, and they rely on like the seawater and the currents. I did research it a little bit, and I did consider making offshore and nearshore one of my measures, but at the end of the day, I haven't really seen much, if any, commercial aquaculture that's actually offshore. It seems like it's mostly in the development stages right now. So I definitely could see that as being like an additional measure in the future, but just for the sake of this work, because there's no commercial species that are being produced offshore, I didn't include it.
Yeah, right now I know in the US they're working on a pilot project in the Gulf of Mexico, but it's been so highly debated, it's hard to know if it'll happen. So anyway, thank you. That's really
super interesting. Thanks for your question.
That's good. I don't see any hand up here. Any more questions?
I think Madison has a question.
Okay. It just came up. Good. Go ahead.
Sorry, I just put it up last minute. Thank you, Annie. This is great, and it's fun to see how this has evolved. I saw a talk of yours a couple of years ago, or a year ago, or whenever that was. So it's really great to see this evolve. I wanted to ask you a little bit more about your how you evaluated those different levels of risk and how you quantified that, and what the basis for those numbers were. If you could just speak a little bit more about the process there.
Definitely. And so I identified the different measures to try to kind of be like universally encompassing. And then when it actually came to calculating the risk, it is not like 100% science, because oftentimes, like I looked at a lot of life cycle assessments and different studies to see what was said. So like it's pretty clear that extensive aquaculture relies on no fish feed, and so therefore the impact on the marine ecosystem and on fish populations, according to fish feed, is 0 or very low. And basically I tried to it's all relative within every measure. And so rather than being like an exact science, as I mentioned, it's based on the relative nature between the different components inside of it, if that makes sense. Yeah.
Thank you so much.
Yeah, that's very good, Madison. It's good you recollected your earlier discussion about the work, right? It's amazing. And once students start and finding your way, and then suddenly it just happens. That was what happened here.
It was the 502. Was it?
Yeah.
Is it called 502? Where
that was the presentation.
Yeah.
Yeah.
Excellent. Yeah. So now Hadi has his hand raised. Over to you, Hadi.
Yeah, I've actually sent you an email about this, Rashid, and it goes back to slide 11, which is the risk characterization slide.
Yeah.
So even though, Annie, you don't want to make this quantifiable in a kind of a strict sense, I think it might be really helpful to spell out all the different dimensions. Because you have multiple dimensions to think about. One of them is, how does feeding these things impact, both internally or externally, land-based ecosystems, ocean fisheries, etc. Another is, how does selection of the species that you raise influence what would happen should the cage break, right? So one of them is dispersive. One of them is potentially invasive, and right? And so in a dispersive system, if you do have high flow rates, then you could expect the dispersive system to have diminishing effects through time. But an invasive system would have growing risks over time, right? Not one, it wouldn't be one, it could be 10. So I really encourage you to kind of just spell it out, even if you don't want to put numbers to it.
Just say.
Characterizing the different risks at this stage would be really useful, because then, through time you can gather more information about whether you can in fact bring them down to one risk scalar instead of a risk vector.
And the only one that I didn't include that I considered including was nearshore and offshore. And you, in my presentation, I didn't include all of the measures, but there is one that's based on if it's like open, closed, or semi-closed, another based on native or exotic. So there is more measures that just aren't included there, but that's a very interesting point regarding the scalar and the magnitudes.
And how they change over time, likely depending on the type and so on.
Yeah,
Very interesting.
it feeds into this whole business of cumulative impact assessments and so on. And if you want to talk about it further, I'd be more than happy to chat about it.
Excellent.
Sure, that would be great.
Good. Great. Any more questions? If not, there's another point to raise in your email, Hadi, about what the subsidy does to the larger economy. And Annie, one of the work you did as of your thesis is doing this multiplier impact assessment, So could you say if we spend $1 to do this, how is it likely to multiply through the economy?
So the total economic impact of the fisheries and aquaculture sectors are about from like 2.5 to 3 approximately is what has been shown in the literature. So that is just another way that whether it's like lessens or exacerbates like the impacts of aquaculture, as subsidies value can kind of like blow up.
Can I just add a point? If you go to slide 17, you will see, or compare it to slide 18, for example, you will see that, if you want to, you can see that the Chinese invested heavily in infrastructure, which would almost entirely go to their own economy, right? But if you go to the next one, Indonesia, or the one after that, assistance, or, you kind of wonder if this money is given to management, who is management? Or if it's given to development, does that stay in the economy of Norway, or does it go to the shareholders of the aquaculture owners, right?
Well, somewhere,
Yeah, but basically, does it actually generate further economic benefits there or not? I mean, if we had a 3 times multiplier in a project in Canada that's subsidized, the tax returns from that would more than pay for it. So as far as the government is concerned, it doesn't cost them anything.
And I did a little bit of reading into the regulations and policies of each country just to understand the context a little bit more. And China has shifted a little bit more towards like sustainable production, but China and Indonesia are a lot more concerned with supporting smaller scale farmers and local food security. They have metrics on like livelihoods and on the income of farmers, how they want that to increase. And so I think you see their motivations reflected a lot in their subsidies and in what they're subsidizing. Yeah.
Very good. Yeah. Yeah. No, this is a really interesting. This just shows that, of course, there's a lot to do. It's just scratching on the surface of this area, really. So, which is what you want to do if you're finishing your master's. You don't want to answer all the questions. So well done. I don't see any questions up there. Anyone wants to say anything here? Okay, I see hands going up, Naveen. So maybe, thank you so much for the presentation. It's been well done.
Thank you, everyone.
Thanks. All right, back to the real chair for this.
Thanks, Rashid. I think we have a break now. Bonnie, don't we have a 15-minute break? And we'll return at 1:30. Is that what the timeline says? Let me look at it. Or were we supposed to return at 1?
Timeline says 1:15.
Oh,
we are running behind.
We're running behind.
Actually behind.
Yeah. Okay, so should we make let's make it a little shorter. Let's make it a 10-minute break and be back here precisely at 1:22.
1:22, all right.
See you guys.
Great, yeah, thank you.
I'll hand it over to Naveen to introduce Ginni for the next talk. Take it over, Naveen.
All right. I'm really pleased to introduce Ginni Braich, who's been doing a PhD with me since 2017. She has a background in plant biology and genetics, got a bachelor's degree from the University of Calgary and a master's degree from the International Max Planck Research School for Molecular Biology. She then worked for Alberta Innovates as a research program manager. It was a provincial funding agency. Then after doing that for about 4 years, she came to do a PhD here at UBC. Her research is looking at crop yield trends in Canada, as well as climate impacts. You may notice that this has actually nothing to do with the title of her talk, which brings me to probably what's most interesting about working with Ginni. She's curious, follows her nose, and gets excited about all kinds of new ideas, and has been working on multiple kind of side projects. So as a supervisor, I'm constantly trying to nudge her back to her PhD, but I've also really enjoyed working with her on various research tangents. At some point, I suppose she'll have to write a thesis with a narrative that ties all of these projects together, but that's another problem for another time. Over to you, Ginni.
Thanks, Naveen. Thanks for that introduction. I'm just going to share my screen here. And can you just give me a thumbs up, Naveen, if you can see everything? Awesome. Great. So, are you able to see the Zoom video, or it's okay?
What do you mean by Zoom video?
You're not able to see your own pictures, are you? Sorry, I have two screens going on, so it's a bit.
No, you're fine. So.
Okay, great. Thank you. So as Naveen mentioned, this is one of the projects of my PhD, and it's a project or a topic that I've been interested in some time, which is GM crops and their use in agricultural systems, particularly to see if they have an impact on yields. So during this talk, I'm going to provide a background on GM crops, talk about some of the trends in their adoption, and as well provide some context on the work that's been completed to date. I'll then go over our rationale for why we're trying to address this question, go into some details about the different methods we're employing to tackle this problem from multiple angles, and then follow that up with some key results from our analysis. So I'll start with a primer on GM crops and kind of the broader context of how crops have been genetically altered to date. So traditional breeding has kind of been the historical way in which we've generated new varieties through the crossing and propagation of plants based usually on physical traits that we desire, so quality and quantity. We want more fruit, we want it to be hardier, but more recent crop modification techniques have been introduced, things like mutagenesis to randomly introduce changes. RNI is another recent invention where you silence genes. And this one is maybe not as widely seen, but you can see it in the non-browning Arctic apple that's currently available in markets. So when we're talking about transgenics, when we're talking about GM crops, we're talking about transgenics, which is how most currently commercial GM crops are modified. And so this is a method where you introduce select genes from other species, hence the word trans, into and that are inserted at a desired location. So some of the most commonly grown varieties use something like the BT gene, which is a gene that expresses a protein from a bacteria which is toxic to insects, or herbicide tolerance. More recent techniques have also included gene editing, which is more precise and uses molecular tools like CRISPR-Cas9. And you'll hear more about the social dimensions of gene editing from the next talk, which Sarah is giving. So first introduced in 1996, GM crops now make up about 191 million hectares. They're grown in 26 countries, and they represent about 4% of global cropland. 99% of GM area is currently grown to 4 crops, so that's soybean, maize, cotton, and canola, but there are other GM crops that are grown in smaller areas. Other vegetables such as eggplant, fiber crops such as poplar, and some ornamental flowers as well. And this figure shows that although industrialized nations kind of made up the early adopters, developing nations now make up a larger share of GM area. And the figure also shows that soybean kind of makes up the largest global share of GM area, followed by maize, cotton, and then canola. And on a crop by crop basis, you can see that 78% of global soybean area is grown to, global soybean area is grown to GM soybean. Similar for cotton, but smaller amounts for maize and canola. So when discussing GM crops, genetic modification is referred to, or a new genetic modification is referred to as a new GM event. And this figure illustrates that most events that have taken place to date have predominantly either been for insect resistance or herbicide tolerance, with much fewer modifications for things like product quality, pollination, and very few events that are specifically targeting yield. So instead, we could argue that most GM modifications have largely been either to increase the convenience or the profit of farmers through these focusing on agronomic traits. Part of that is also the ease for modification. One is a simple trait such as insect resistance and herbicide tolerance. Traits such as yield are a lot more multifactorial and there's no one yield gene, so it requires a lot more precision in editing than has been historically possible. So why bother with the yields of GM crops? Why wade into these murky contested waters? You know, as we can see from the trait data, genetic modification has not directly been to improve yields. However, there are a lot of yield benefits that could be realized through reduced losses, particularly in places that might have lower input use. And of course, as we know, yields are not not necessarily the most important outcome of an agriculture system, but they are key to farmer profits and an important component of the land sparing versus sharing debate. And as we can see from the uptake data that I'm going to show, farmers are clearly seeing some benefit from GM crops given their large uptake despite there being higher costs in conventional seeds. So our argument is that a more thorough analysis on the subject could provide some clarity on the topic. And as I'm going to show, the analysis of yields to date has not particularly been nuanced. So what has the discourse on yields been? So this is the article from the New York Times in 2013 about the promise of GM not being fulfilled, and it uses graphical comparisons between some cherry-picked data from the US and Western Europe on yields and input use. And they do provide some interesting comparisons for yield, but this doesn't account for differences in growing season conditions and other country level yields. And the input data that they use also doesn't, it's not in the same units, it's not for normalized data. So when you actually do that, you can see that, yes, for example, France is reducing their pesticide use, but it was a little much higher than the US to begin with. So that kind of nuance is missing at times. There was another recent paper in 2020 in Nature Plants, which looked at the impacts of BT cotton from Cranthea and Stone. And again, this just used graphical comparisons of GM adoption yields and fertilizer. And in the paper, they allege that improvements in yields have been due to fertilizer and not to GM, but they didn't try to actually attribute the rates of yield increase to these different factors. And then on the other side of the spectrum, we have this analysis from Klumper and Quain in 2013, where they looked at 147 papers on GM crops and find that GM had yield increases of approximately 22%. There's some nuance again missing from this paper because they're relying on published studies, which is largely based on experimental plot work or a farm survey, so it might not be an accurate representation of what an average farmer's performance is with these seeds and with these chemicals. And again, there's no breakdown by crop. So given all this work to date, if we wanted to approach this question, what are the sort of kind of things and nuances or confounders that we need to consider? And some of those are, we need to consider the temporal and spatial variation for different countries when it comes to climate, irrigation, fertilizer, their herbicide and pesticide use, but also their differences in agriculture systems, countries having different soils, means of production, infrastructure. And of course, there being time trends, so a lot of other technology improvements such as improved varieties, better equipment and management practice as well. And then we can try different methods and see if we arrive at the same result. So to answer these questions, we assembled some panel data. So from FAO stat, we got country level yields, fertilizer, irrigation, and pesticide use. Unfortunately, except for yields, these are all at national level and not crop specific. We also got national climate data from for temperature and SPI, which is a drought index. And lastly, we got GM adoption data from the annual reports of the International Service for the Acquisition of Agri-Biotech Applications. So this is an organization that collates information on GM technologies. And so we have a data set of GM adopting and not adopting countries. So in terms of the countries we focus on, although I said there were 26 that are currently growing GM crops, we focus on a much smaller amount because we
lost connection, maybe.
Looks like Ginni is frozen. Okay. Let me. I don't know if I have her
an email and ask her to re-log in.
Yeah. Do we? I can message her.
I'm just going to try calling her.
Yep.
Ginni, are you back? I heard her momentarily.
I heard her, yeah.
Kushank, do you mind calling her?
Yeah, I'm just calling her.
Twice in one meeting. We're doing well.
Suspicious almost.
Hope it's not Zoom giving up on all the load. I don't know. It's crazy.
Hasn't usually been this bad.
Yeah.
She's still not back.
Maybe it's internet.
She had a problem with her internet. She's just connecting back.
All right.
Okay.
Good. It's not Zoom. That's nice.
She's back.
So sorry, my internet just cut out. Can you hear me?
Yeah, you can hear you, but you're still cutting in and out.
Oh dear.
Maybe you should cut off your video and see if that works.
Yeah, maybe cut off your video and see that helps. That's a good idea. Ginni? Maybe cut off your video. Or are you frozen again?
Are you able to?
I'm afraid you're still frozen.
I wonder if we should all cut off our videos until discussion.
It looks like most of us are, except me. So
Funny, I only have this trouble with seminar series. I can teach a class this size and don't have that trouble.
okay, Ginni, do you want to try saying something? Your video is now cut off. We can see your slide, but we can't hear you. Oh boy. I think we lost her again.
Very frustrating, isn't it?
Yeah, she's completely out again.
For you to get the presentation and for you to do the screen sharing and she can talk.
Oh, I thought you were asking me to do the talk.
Yeah, no, that's not.
Say no, thank you.
No, but if you could get her presentation and then share your screen, it'd save her bandwidth from transmitting that part of it.
Yeah, Kushan, can you call
Here
her?
she
I
comes.
don't seem to have enough. That's a good idea,
yeah.
Kai.
Is that better? Sorry, this has been a disaster.
No worries. Kai just suggested. Sorry.
I was saying maybe to give her time to adjust. Okay, she's back.
I seem to be back. I'm sorry, there's been Shaw outages the last 2 weeks and we thought we had it figured out, but apparently not.
You're now back clear. So if you want to just keep going, that's fine.
Okay,
Kai
I'll try
had
to-
an alternate. Yeah, just before, if this keeps happening, Kai had a suggestion that maybe you could send me your slides and I could do the slide sharing for you, but let's keep going.
Okay, let's see if that happens.
Do you want to go one slide back just so people remember where you are?
Did you guys hear where I was discussing about the different countries?
Yeah.
Okay, so as you can see, North America, South America have the highest number of GM adopting countries. If we look at GM adoption rates, you can see what percentage of a national area of a country was planted to a GM variety. And this is from 1996 to 2018, and you can see that there's been pretty rapid adoption of across most crops and across most countries, with almost all countries having at least 50% adoption by 2010. Brazil is particularly interesting because it was a relatively late entrant and is now one of the largest growers of GM crops. And there are still some countries in the data set that have had lower adoption rates, such as Spain for GM maize and also Mexico for GM soybean. So with all this context now and the data assembled, we can put forth the following question: has GM crop adoption had an impact on country level yields? And we can try to address this question through a couple different methods. And I'm going to go over two particular methods today, which is propensity scoring and also different types of multivariate regression. And both these approaches try to attempt to estimate the effect of a treatment while accounting for potential co-founders that might also have an impact on yields. So with propensity scoring, what we're trying to do is that when we have an experiment, we try to randomly assign treatment or control. However, in a lot of real world scenarios, that's not necessarily the perfect experimental conditions don't exist, either for ethical or practical reasons, such as when we're looking at the health outcomes of smokers versus non-smokers, student achievement in public versus private schools, or the GM yields of adopting versus the yields of GM adopting or non-adopting countries. Instead, what we can do is try to set up a quasi-experimental condition to make our treatment and control group as balanced as possible. So what we mean by this is that when we're looking at the effect of GM on yields, we want to compare apples to apples. So through propensity score matching, we can estimate the effect of a treatment by accounting for all the covariates that predict receiving treatment. So what this does is that if there's a bias that arises because a factor that predicts treatment may be the reason for a difference between treatment outcomes, we can try to control for that. So in the case of GM, it could be that wealthy nations are more likely to adopt GM, and their country's wealth is somehow tied to their higher yields rather than the fact that they're growing GM. So this just gives a nice little visualization of how propensity scoring works. So we can assign everyone in our population a score based on some baseline characteristics, so how much their GDP per capita is, what their climate's like, their fertilizer use, and so that countries that are similar are being compared to each other. So it's not like we're comparing the maize yields of the US and Benin and arguing that the higher yields in the US are due to GM and not a slew of other factors. So what propensity scoring does is after we've assigned a propensity score for each country, we can then match based on different algorithms. And so that's what we are seeing here is that we have propensity scoring of every country in our sample before matching and after matching. The one downside, as you can see, is that there are control units and also treatment units that go unmatched. So that is unfortunately one of the drawbacks of this scenario. And then we can try different methods. So one approach is to use a binary scoring. So we're saying countries that have at least 50% GM adoption are a treatment country, and those that have none are a control country. And what we see is that the yield difference between GM adopting and non-adopting countries is positive for all crops. And we have 95% bootstrapped confidence intervals. And so we can see this positive result ranges from 19% for soybean, 21% for cotton, and 51% with very large intervals for maize. Another approach we can take is to use a continuous treatment. So we're using our actual GM adoption rates and not whether a country has 50% GM adoption or not. So this approach uses weighing rather than matching, so we're not losing any control or treatment subjects. But again, you can see it ends with all the covariates being balanced. So there should be no difference between our treatment and control groups when it comes to all these other confounding factors. And using this approach, we can see that for every 1% increase in GM adoption, yields on average for soybean increased by 0.16%, for maize 0.89, again with very wide confidence intervals, and for cotton 0.52%. And so if we had 100% GM adoption rate in a country for cotton, for example, we could say potentially, according to this method, that those countries would have 52% higher yield because they have GM. Another approach is to use more traditional multivariate regression models. So again, this is trying to control for all these other confounding variables to detect if there's an impact of GM on yields. And all of these approaches allow us to account for that noise from input use, weather. But unlike propensity scoring, it also allows us to account for differences between countries and time, which is not considered in propensity scoring. So if we have differences between countries, we can include those as a fixed effect. So we can control a lot of time invariant factors such as soil quality, or we could have every country have its own intercept through something like a mixed effect model. And we can also account for year effects. So we can account for elements that might change over time, but are common across locations. So things like the rate of non-GM technological improvement, management practices, any widespread climate events. But in other types of models, we can also allow each country to have its own time trend if we think some countries might be developing more quickly than others. And in addition to that, we can allow our time trends to have different functional forms if we think time is in a linear fashion or a quadratic fashion. And lastly, this approach allows us to use more robust approaches so we can see the influence of outliers or influential observations on our analysis. So I'm just going to give a simplified example of one such modeling approach, and that's difference in differences. And what this does is that it looks at the difference in changes of yield over time that occur between adopting and non-adopting countries. So you can compare early adopters and late adopters, but also early adopters' yields before they adopted and after they adopted, as well as between non-adopters. And so what this allows is for us to account for heterogeneity between countries, treatment timing, and also amount of treatment. So I know this is quite a busy figure, but what it allows is for us to see a lot of sensitivity analysis in one place. And so under the models, what you can see is that we've tried multiple different models ranging from simple linear to robust linear mixed effects. We've tried different time trends. So time being linear, quadratic, cubic, or potentially even logarithmic. And then we have our three types of crops, so cotton, maize, and soybean. And as I mentioned, all these models make different assumptions about how the effects of different countries and time are treated, as well as to whether the analysis is robust to outliers. And from these models, we've also included all the confounding variables such as GDP per capita, fertilizer, pesticides, etc. And as well, I've reported the root mean square error of each model. So this gives an indication of the spread of the residuals, or rather how concentrated the data is around the fitted line. And so from this figure, we can see that for every 1% increase in GM adoption, yields on average for cotton increase by about 0.3%. For maize, it's about 0.2, but as you can see, very sensitive to a time trend. And for soybean, it seems to not cross the threshold of 0 in a significant way. So if we look at a comparison of our approaches, we can see that for GM cotton, it seems to indicate that they have higher yields with higher rates of GM. And there is an overlap with the two approaches that we've taken, either the continuous propensity scoring or the different multivariate regressions. The results are less clear for maize. The regression approach shows that the results are highly sensitive to a time trend, and the range is very different compared to the propensity scoring approach. And for soybean, we can see that the regression approaches demonstrates almost no effect of GM adoption on rates on yield, given the different models and time trends. And this is in somewhat of a contrast to what we find from the propensity scorings, where it shows a slight positive effect at least. And we can also contrast this to the findings from the Clumper and Quain meta-analysis, which found about 21%. And this, of course, is assuming 100% GM adoption because they looked at either adopting or non-adopting trials. And as I mentioned, they don't separate out by crop and use this field trial data. So maybe it's not a perfect comparison, but it shows that maybe our cotton estimates are within that range. So just to get to some conclusions, for maize and especially for soybean, it seems like our results were especially sensitive to the approach used and some of the modeling assumptions made. However, in contrast, the trends of GM cotton seem to be pretty robust either to approach and also to some modeling assumptions. So I think this does demonstrate that there is a need for multiple approaches and robust methods when working with this type of data, especially because there are so many confounders that influence how yields increase over time. And I think when we talk about the implications for GM, it remains a nuanced story, but this analysis might help clarify one aspect of the story, which is that we can clearly see that GM cotton has improved yields. And as I mentioned, there does seem to be some benefit to farmers, but none of this analysis is really capturing any environmental impacts. So I'm sorry again for the interruptions. I just want to give a particular thanks to Naveen, my supervisor, and also to Zia and Frederic, provided a lot of insight and advice on this project, and lastly to the Luge Lab for all their ideas and encouragement. Thank you.
Thanks, Ginni. If you want, maybe try to turn your video back on. See if that works.
Let's hope.
All right, Milind, if it's okay with you, we'll take 5 to 10 minutes for questions, unless you think we should move on. Don't see a hand up yet. Rashid, maybe we'll go with Harold first, since we take students first. Harold, and then Rashid.
Yeah, okay. Yeah, I came in because there was no hands up. You can ignore.
Thank you.
Thanks for that great talk. I was curious that 0.3%, is that, how much is that relative to the other sorts of practices that are used to increase yield, particularly in cotton or in these other crops? Is this, regardless if it's positive or negative, but is this actually a significant amount as far as yields are concerned? Or is this really kind of meaningless relative to other methods that we have to increase yields?
So I can give a bit of context with traditional breeding. So right now, when breeders are breeding for cotton or maize, they're lucky if they get around a 1% increase per year with a new variety coming out annually. But when we're looking at these GM percentages, so that's 1% for every, potentially 1% or 0.3% for every 1% increase in GM adoption. And like I showed, most countries are at about 75% threshold or higher for their GM adoption rates. So then if you compound that up, that's about a 20% increase in yields compared to a non-adopter when you control for all the other factors, including time. So potentially it could be a big impact, but there are a lot of improvements that happen through traditional breeding programs that GM alone can't capture. So when we're looking at abiotic stress, breeders are also breeding for quality parameters, and that's not necessarily the case for GM crops, which is one of the reasons why Burkina Faso actually abandoned GM cotton, is that they weren't getting the quality characteristics that they were used to, despite getting higher yields. I hope that helps.
Rashid, do you want to go? I don't see any other hands up.
Do you have time, because of there's one more speaker, right? I don't know.
Yeah.
Yeah, so just go ahead. Go ahead. I just wanted to feel the space, the time, because there was.
Okay. All right. So Linda's back to introduce Sara.
I think that's on
Terry,
my
can you do
docket.
that?
Yep, I can do that. And I'll let Sara load her slides, but Ginny, in a way, that talk is a perfect entry into Sara's work. But Sara, do you want to share your slides first? And I'll just say, while she's loading up her slides, that Sara has done an incredible amount of work putting this thesis together across many different kinds of efforts, as she's just tried to sort of pull apart the puzzle of broadly stated public response to genetic engineering, looking at that from, why policies do or do not trigger regulation on this count, what the big debate is in global fora that she'll tell you about, how people are making sense of these things, and how well is that captured in the sort of discourse on that or not. Also, Sara, I want to just thank Melinda and Shannon Hagerman, who's not here, for heavy lifting on this thesis work too, but and also just to nod to the sheer pleasure of working with Sara's curiosity and kind of voracious appetite for research. Just been a fabulous journey so far. So Sara, I'll turn it over to you.
Sure, thanks so much, Terry. Yeah, I also feel like I need to, can you all see my screen, by the way?
We can, yeah. And it's in the right mode.
I just also wanted to kind of second what was said earlier about, just how wonderful Terry has been as a supervisor and mentor. So she's not going to like it that I'm
Yeah, no, she's not, but change the subject, keep going.
Yeah, and it's really great to follow after Ginni as well. I think it's a good segue. So I wanted to talk today about try to talk about my PhD project as a whole, which has focused on the social dimensions of gene editing technologies in agriculture. And I thought to introduce gene editing, Ginni has kind of already got us started with this, but I thought it'd be useful to place it in the context of other ways of altering or modifying genes. So we have conventional breeding, which often involves crossing different organisms in order to obtain desirable traits. We have genetic modification, which usually refers to the use of recombinant DNA technology to introduce new genetic material, usually from an unrelated species into an other organism's genome. And then we have an important new host of techniques, which is gene editing. And that generally involves, like GM, cutting an organism's DNA, but here the cuts are at more targeted locations, at least in theory. And sometimes they involve just smaller changes, like very short deletions. And CRISPR is the best known technique for gene editing, but it's not the only one. So proponents of gene editing are heralding these as different from GM and more like conventional breeding, essential for addressing climate change and food security. And they're also arguing that due to its precision and low cost, gene editing is democratizing biotech, facilitating new ways of using it, such as for smallholders. However, these technologies also offer a lot of new risks, ethical dilemmas, and challenges for governance. So to say that GM debates, both public and scholarly, have been divided would really be quite an understatement. And an important non-technical concern that's often debated is that of naturalness. So there's been a tendency to dismiss concerns of naturalness as irrational. For example, you have GM proponents criticizing those that call GM a frankenfood or claim that it messes with nature. And when I began studying this subject, it really struck me that gene editing is seeming to kind of open the door to a pretty vast new set of biological possibilities. And as a result, that it might be generating concerns around naturalness, but also perhaps even shifting what we even mean when we talk about naturalness. So a question I wanted to ask across the various chapters of my thesis was, to what extent are concepts of naturalness useful for thinking about the social, ethical, and governance implications of gene editing technologies? And the first project I'll talk about tried to take stock of different ways of thinking about the naturalness of food and landscapes in particular. And I wanted to ask, what might be some ways scholars have thought about naturalness, and how might we assess which of these are useful for studying people's thinking about gene editing? So to do this, I drew upon work by agricultural and environmental ethicists to create a framework of logics of naturalness. I then reviewed definitions of naturalness used in a range of empirical fields. And then I applied the original framework to a set of example cases of gene editing with the goal of evaluating which logics were most relevant to how people might actually think. So to give a bit of a flavor of this, we can take the case of microorganisms that have been edited to have enhanced nitrogen fixation capabilities. And I theorize that some may find this unnatural simply because it involves direct modification to the genome by humans. It's also possible that others might see it as natural, as it arguably involves supporting an organism to perform a function that it was already performing to begin with. And others perhaps might see it as natural because it doesn't involve an organism that most consumers have a particularly strong personal relationship with. And so on. And if this example is already raising a number of questions for you, you're not alone, and you can perhaps also see why a main takeaway from this project for me was really just how difficult it is to pin down ways people might think about naturalness. And developing some all-inclusive theory of naturalness really feels quite like an impossible task. But I think another takeaway is that delving into reasonings around the concept has, I think, the potential to help us begin thinking less about naturalness as an irrational response, as has been the tendency, and more about which values might underpin concerns about naturalness, such as values of ecological harmony, about social or spiritual relationships, and so forth. So the next project with Melinda explored regulation of gene-edited crops. And we first reviewed whether and which types of gene-edited crops currently trigger biosafety regulation in different national jurisdictions. And we also asked whether that approach seems to be working or not, and if not, what a better approach might be to regulating crop gene editing. So our review found a few themes. First, a common criterion across most jurisdictions was that something would be regulated on the basis of whether it contains novel genetic material. So an example of this is the EU's definition that it applies to gene-edited crops, which it defines as any organism whose genetic material has been altered in a way that does not occur naturally by mating and/or natural recombination. And beyond this criterion, there were numerous ways that countries draw the line between what is regulated or not. For example, some regulate an organism if a gene has just been deleted, others if only a very short sequence has been inserted, and others if some foreign material is only inserted temporarily in the editing process. And furthermore, there is little clarification in most jurisdictions of whether epigenetic techniques are regulated. So that's techniques where the DNA has not been changed, but other parts of genetic material attaching to the DNA have been altered. So to summarize, we found that most jurisdictions are still really thinking along a modified or not kind of dichotomy, sometimes a natural or not dichotomy. And yet we think this approach is kind of increasingly out of touch with the multiplicity of different levels and types of modification that are possible today. And we also offered a few suggestions for regulating gene-edited crops in ways that are more in alignment with principles of responsible research and innovation. So we suggest a multi-level tiered approach to regulation. So as opposed to having only two levels of regulated or not, and also the incorporation of just a range of broader social and ecological considerations so that the focus is not just on technical questions like around the magnitude of an edit. The next topic I explored was debates that have been occurring at international fora around genetic resources. So as genomics has taken off, a side effect of that is that it's really threatening the legitimacy of international agreements that establish systems of access and benefit sharing. So these obtain consent and provide compensation for commercial use of plants, animals, and microorganisms, particularly those that are sourced in the global south. And so far, these agreements have governed these resources in their physical form. But now research is increasingly utilizing genomic sequences and associated information. So for example, if a company took a specimen of a particular crop species, brought it home and utilized some aspect of it to produce a product, such as via gene editing, that would be covered by benefit sharing regulations. So that company would have to compensate the country they took the organism from. However, if they just use the genetic code downloaded from a database online, right now it is unclear whether those benefit sharing obligations would apply. And that's what's being hashed out right now. Debates over this are really heated, with some arguing that this phenomenon is biopiracy, and others arguing that this information, which is called digital sequence information, or DSI, is essential to research that supports biodiversity conservation and food security. So I wanted to ask, what are the conceptualizations of genetic resources on each side of this debate? And based on my review of formal proceedings and documents, I found two main viewpoints on DSI. So the first tends to be expressed by the global north. And in this view, genetic material is strictly the physical objects, such as seeds. DSI is not considered to be a genetic resource. And also DSI is seen as kind of intrinsically unnatural. We also found that it is the cognitive efforts of researchers in this view that is really seen as generating DSI's value. The view characterized by the global south is that genetic material is not just physical objects, but it's also data and information. DSI is seen to be a genetic resource, and in this view, it's something that is DSI is something that's natural because it's just considered to be part of the physical object. And it's in this view, it's the historical stewardship of resources across many years and generations that's really seen as generating the value. So a key finding from this project was that questions of value, such as when it's added, where exactly it lies, how it gets added, really have defined differences in the debate. And we also found that ideas of what counts as a resource, including what counts as natural, have important justice implications as they influence who gets compensated for the use of genomic data and information. So moving on for my next project, the goal was to try to understand what exactly shapes how people think about specific applications of gene editing. And the motivation for this is that while attitudes towards GM have been quite extensively studied, there isn't much evidence as to whether people will indeed think about gene editing similarly to GM or not. So we asked whether participants' patterns of thinking follow particular archetypes. And we used a methodology called Q method, which is a sort of hybrid qualitative quantitative approach that's really helpful for extracting patterns of thinking on a subject across a population via structured interviews that can be analyzed statistically. And the analysis revealed 3 ways of thinking about gene editing. So we called the first archetype critical system thinking, and participants in this group really felt that the applications in question were avoidable, unnecessary, and that they perpetuated an unsustainable system. The next archetype of pragmatic techno-optimism, folks felt that we are really changing, we are already changing crops and animals to suit our needs, and this really isn't that different than that. And then in the ambivalent questioning archetype, they felt that really just there are too many unknowns to be sure about any of these applications in question. So an important finding from this work was that none of the archetypes really focused on technical specifics as particularly important, despite what many proponents have argued. Second, the critical system thinking archetype also suggests that perhaps what matters to people is less questions of how unnatural a technology is, but rather whether the technology has alternatives or whether it's viewed as necessary or not. And we suggest that this is an important dimension of attitude that might be explored with other groups. So building off this, the next project involved an online survey of people in the US and Canada regarding their perceptions of gene editing in agriculture. And the goal with this survey was twofold. First, the quantitative perceptions literature on GM really doesn't give attention to some of these more system level critiques, such as those articulated by participants in the Q study. And we wanted to understand then whether opposition to or support for applications of gene editing might be predicted by attitudes critiquing agricultural systems more broadly. Second, we wanted to understand how framing gene editing in the context of specific trade-offs might affect participants' responses, such as trade-offs between gene editing and biodiversity loss and gene editing and pesticide use. And the goal here was to understand to what extent people might be open to making this kind of a difficult trade-off, and to what extent they might simply refuse to make that kind of a choice. So we found that while participants were quite open to gene editing when presented in the context of, many participants were quite open to gene editing when presented in the context of specific trade-offs, a large proportion, approximately a third of participants actually opted out entirely of making that kind of a choice. We also created and tested a novel attitudinal scale on attitudes towards the green revolution, with the goal of using it as a proxy for attitudes towards food systems more generally. And we found that alongside conventional predictors like trust, both a sense of the Green Revolution's necessity and also a sense of critiques of the Green Revolution did predict overall comfort with gene editing. And furthermore, we found that attitudes toward the Green Revolution also predicted the likelihood of opting out of the trade-off, with those who were critical of the Green Revolution and those who found it not necessary as more likely to opt out of the trade-off entirely.
Sara, you have 5 minutes remaining.
So our finding around the large number of participants opting out of gene editing begs the question if perhaps those opting out or expressing moral outrage or opposition to gene editing may have reasonings rooted in particular system critical complaints regarding gene editing rather than more disgust-based, affective or other sort of knee-jerk reasonings. And this bears further explanation, such as via more experimental designs that can kind of get at the causal relationships between these attitudes. So, okay, I know I've talked about a lot of different projects, but the last one I'm going to mention is sort of a side project that with some friends that ended up turning into kind of a full paper, and I couldn't not talk about it here because it's so relevant. But we were really curious about the organic sector because it has served as a kind of anti-GM stalwart, and we were curious to empirically explore how the sector is actually thinking about gene editing, given that the only existing work on the topic currently focuses on sort of theorizing about whether organic is theoretically compatible with gene editing. And our question was about how actors in the sector are responding to gene editing. So we found that all actors really agreed strongly that the sector needs to kind of draw a line excluding gene editing, and most felt pretty clear that the two are incompatible. There were a diversity of reasonings for this, which included some who felt more philosophically that sort of any modification with the cell wall interferes with the integrity of organisms. We also found folks who were much more pragmatically concerned around messaging with consumers and sort of the need to assuage their concerns. But I thought a really interesting finding was that there was some intellectual curiosity about gene editing amongst plant breeders. So for example, one wondered, what if with CRISPR we were able to develop a gene to, protect tomatoes through late blight? So now growers don't have to spray copper, which is something allowed in organic, but is not something we really want in our food. So I always think about these things, but knowing that, we still have to draw a line. So we found that the gene editing is kind of forcing the organic sector to do a lot of work to clarify which breeding practices are acceptable or natural, which is kind of opening up a bit of a can of worms, not just around gene editing, but around new practices and also older breeding practices, which are now really kind of beginning to look a lot less in alignment with the sector's values. And also, as a result, the sector is struggling to keep up with the burden of responding to questions about what is acceptable or not, which takes a lot of resources and is shifting the focus away from the sector really being able to enact its principles that it wants to stand for. So a final few reflections on this broader question I had posed at the beginning around whether naturalness is a useful idea or whether we've kind of moved beyond it with regards to studying the social dimensions of gene editing in agriculture. First, I think gene editing is upending existing regulations and standards based in dichotomies. I think second, we're seeing that the technical complexity and variety demands that regulators or standard setters draw lines and boundaries, and yet that task is so demanding that it can make it quite hard to focus on broader questions of value and purpose, as well as more holistic types of implications. Third, whether data is considered to be something that's natural or not has pretty important implications for global justice. As well, system critical attitudes, I think, can offer potentially more useful dimensions of attitudes towards gene editing beyond conventional measures. And last, kind of getting back to the first chapter, despite how slippery the concept of naturalness is, my sense is that it's a concept that's still worth engaging with. And I'm curious if attempting to define and classify understandings of naturalness might help to highlight some of these deeper value-based rationales that may underpin what can seem like more rational impressions or feelings. That's all I have. Thanks so much.
Thanks so much, Sara. Really fabulous cohesion across those many and seemingly disparate efforts, but came together really well. And lots of kindred spirits on topics here for Sara and for everyone here, those working across food, across risk, across biodiversity. So I think she'd probably be up for any number of questions in that huge umbrella of things. Yes, and clapping popping up as well. Questions? Should I jump to Naveen, since you've got your hand up and we have limited time?
Happy to let a student go.
I don't see one yet.
Okay, I actually don't have much of a question, but more of a comment maybe. Lovely work, by the way. It was an amazing set of 6 projects, and 6, that's pretty amazing too. About your 4th project, the one where you looked at system critical conversations. I have a sense that that's true. I think Nathaniel Johnston in Grist has a series. I don't know if you've read those on GMOs. Oh, yeah. Yeah, it's some of my favorites. It was one of my favorite series when I was trying to understand GMOs. And so he goes through this whole series of, I think it's something like 13 articles, and he concludes in the end that the argument about GMs are a proxy for a whole bunch of other things, that it's not really about GM, but it's about system critical debate. So that really jives with what your fourth project is saying too. Anyway, from a question perspective, I think my question would be then, how do we navigate? How do you take this understanding and navigate this going forward, I suppose?
Yeah, thanks, Naveen. I think with regards to the system critical thing, I wanted to mention an interesting finding was that we also included in the survey, along with sort of this Green Revolution attitudinal scale, a scale on corporate attitudes towards corporations, basically. And we found that was not as predictive as this Green Revolution scale. So I think it's very easy to kind of default to just broader sort of political critiques, but there does seem to be potentially something really interesting here around visions of agriculture and what that system should look like. That's different from I don't know, maybe it's just that everyone's now critical of corporations, so it's no longer a useful metric. Mm-hmm. But with regards to how we move forward, oh gosh, so many things. I mean, I think that it would be really interesting to study some of these system critical attitudes a bit more, just because this is it feels like just a first stab at it. And particularly something I'm sort of kicking myself over is not including more of a measure of naturalness in the survey that we did. I think at that point in my PhD, I was very frustrated with the concept and felt like it was a bit useless. So I didn't make it into the survey, but I think it would be really fascinating to sort of have that comparison, or that possibility of comparing their relative power, or perhaps seeing the relationship between the two kind of concepts, especially if you theorized naturalness or developed some sort of attitudinal scale based on some of the logics that I sort of summarized in my first chapter.
That eternal regret with surveys. That was one though. Maybe I'll jump to Juan Diego before Hadi and Milind, since he's one of the students with their hand up.
Yeah, so thanks, Sara. Amazing work on a fascinating topic. I wanted to ask you about, I think was the third project on the perception of, how the global North and the global South perceive like DSI differently. And I mean, it's expected based on profitability, but also based on the biodiversity of what's in the global North and what's in the global South. So like, I was wondering what's the current state of the discourse there and the negotiations, and maybe a clarification question on who was it that you, these were scientific papers or these were regulatory reports? Because yeah, I'm just wondering whether it changes from policymakers who have like a strong political agenda versus just lab researchers studying genomes.
Oh, thanks, Juan Diego. So the documents I used were all basically the Convention on Biological Diversity and the Food and Agriculture Organization of the UN's Plant Treaty. For each of those, there's a set of like formal UN meetings, and for each of those sets of meetings, there's a series of papers, documents that get put forward that debate these issues. Those include kind of reports that each of those institutions will conduct, where they go off and survey a number of people. They'll ask for comments from any country, any party to those agreements or conventions or treaties that want to comment. And they just publish all of those on their website. And so I just went through all of those and found ones that kind of where this topic was being debated over the past 7 years or something like that. So that's kind of the raw material that I then went and coded qualitatively and kind of extracted some of these key viewpoints from. And in terms of where the debate is now, it's really kind of stagnated. And I think that the default position is that digital sequence information, which is incidentally a very controversial term, but I'm calling it DSI just because it's easier to have one term. But it's the default position is that DSI is not considered a genetic resource. So only the physical material is kind of that's sort of where you sort of get to in the face of this kind of stagnated debate. So yeah, I don't know if that fully answered your question on where things are at right now, or if you were asking something different.
Yeah, no, it does. And yeah, I'm just wondering whether a deeper look into what policymakers and like people who usually speak at this fora think, contrasting to just researchers. Thanks,
Yeah.
Sarah.
Time for one more, Hadi. It's 2:17.
Yeah, I'll be quick. First of all, that was a great talk. Thank you very much. So well connected, etc. I want to follow up on Juan's thing. So, how much of the DSI is junk information, right? And the differentiation between the junk information. Well, first of all, what we call junk could not be junk, but we don't know whether it's junk or not. The other is that in gene editing, it's very much about the functional part of a sequence. And so I was wondering if you wanted to talk about whether in your interviews and discussions, you have run across people who are knowledgeably talking about these things rather than emotionally reacting to these things.
Yeah, I mean, I guess I would maybe push back a bit and say that I think that what's so challenging about this topic or this debate is that I don't personally think there's like a right answer necessarily, in the sense that it's quite a philosophical question of is like the information that you extract. So, okay, so you have something like maybe the actual data, sort of the sequence itself, which certainly you can make philosophical arguments about whether that is something that exists separate to or inherently part of the object. You also have things like annotations, phenotypical annotations that people are sort of attaching to sequence data, which is, I think, more arguably separate from the physical object itself. But I really do think that initial question is quite a hard one to get at, in terms of it's hard for me to make a firm argument about whether it is or is not part of it. Yeah.
You know what it reminds me of is NFTs and art. If I were to take a picture of if I take a picture of Mona Lisa at 3,000 dots per inch with 24 bits of color depth, right? It would look exactly like whatever it is in the Louvre. It still wouldn't have the texture that I would get it, right? But if I were to degrade that image by 90%, it would still be identifiable, a picture of Mona Lisa, right? And so the whole question is, if somebody came along and said, what is this? Could I claim that it's not Mona Lisa's image? Anyway, so it's a really interesting question, but I do think that we have rules in copyright that clarify these issues, and it seems that people in the North are trying to have it both ways. They want to have their copyright, but they don't want to offer copyright protection.
Yes.
Yes.
Yeah, that's a great analogy, Hadi. So Milind, I'll turn it over to you for closing question and closing master of ceremonies.
Oh, I didn't know I was the MC, but I'm
you are now, and we're.
So you know, Sarah, the whole sort of system critical stuff. I mean, years ago, when I was traveling around India talking to people, it was the diversity of reasons for why people oppose GM just was flabbergasting, right? And the only analogy I could think of was the boggart. I've said that 100 times in all my classes. The boggart in Harry Potter is transgenics. I mean, just about everything. The Gandhians hated for Gandhian reasons. The anti-pesticide folks hated for anti-pesticidal reasons. The anti-globalization folks hated for anti-globalization reasons. I mean, you name it, we can find a category to fit it, right? Now, what about, so I was interested that there were these organic agriculturalists who are sort of seeing potential benefits. What's going on? Are the anti-GMO, is it as a demographic turnover, or just people dying and being replaced by new people? It seems to me that's the only way I can see gene editing taking off. It's just a comment.
You mean amongst the organic plant breeders, organic sector? Yeah, I think just to highlight again, it was the plant breeders particularly who were kind of expressing that curiosity, which I think makes a lot of sense if you think about them having more of like a natural sciences background and coming at the topic from a slightly different position than others in the sector that we talked to.
Okay, so not what I thought it might be. Thanks. Well, thanks a lot, Sara. That was fantastic. That's just a really awesome presentation, great work, sort of amazing oeuvre spread across so many different interesting projects. Well done. And this is to all speakers too. Ginni, Gurneet, did I get it right, Ginni? Gurneet, and Annie, thank you very much. This was brilliant. This is one of my favorite IRES symposia, and maybe it's the format. I don't know, the Zoom format. People asking questions on the side, etc., etc. I'm sorry I said that, but anyway, we'll all next year hopefully have it in person. Thank you very much.
Thank you, Milind. Thank you, everybody. What a pleasure.
Absolutely fantastic. Thank you.
Thanks, everyone.
Want to stay on for a second, Sara? Or Bonnie might kick us off.
No, I said I'd leave it on for a couple minutes.
Oh,
if
okay,
Sara
great,
wants to read
Bonnie.
the chat feedback, because we lost it for another speaker before.
Or I'll
That was awesome, Sara.
Hi, Maddie.
Take off,
It was
but-
great. Okay, see you later. I'll tell you what, Sara, I was just before we started, I was writing you a long email about other stuff. Maybe I'll send you a Zoom link quickly now. We'll just drop out of this one. Have you got 5 minutes? Okay, all right, talk in a bit.